For this week, let's talk about the microscope. So, besides the lecture video, I also posted some links from YouTube regarding the microscope so that you can see the animated version of the parts of the microscope. Kung paano ba yung kanyang mga function and all. Since we do not have a microscope. Dapat kasi, when I discuss this topic, usually nagdadala ako ng microscope sa class and then yung mga estudyante, ginagamit nila yung microscope just for them to see and feel the microscope. Okay? So, let's start regarding microscopy. When you say microscopy, this is the process that involves the following. So, first of all is the magnification of objects. So, as we all know, sa ating practice, we deal with microorganisms or microscopic organisms. And then, it is very important for us to visualize those organisms or microorganisms in order for us to see kung meron ba talagang presence or absence of that microorganism. Okay? Kasi nga naman kapag may microorganism, that can signify an infection. So, hindi lang naman siya para sa mga microorganism, pwede rin sa examination of cells. So, just like examination the cell if the cell has cancer, diba? For cancer diagnosis, tinitingnan natin ang mga tissues. If you see abnormal tissue pattern or mutations na nakikita, then may kita mo yun sa mga tissue talaga under the microscope. So, you can now tell that the patient has cancer or wala. So in our practice, we have different types of microscope na common na ginagamit. May mga iba pang klase pero hindi naman common sa practice natin. Pero ito lang talaga yung common. So you have the bright field or the light field microscope, also known as the compound microscope. Okay. The fluorescence microscope, phase contrast, dark field microscope, and also the electron microscope. So yung bright field na yan, ang common na tawag din dyan is the compound microscope. So, let's first talk about the light bright field microscope. So, as you can see, meron tayong microscope sa picture but meron akong video na posted dyan sa bandang baba ng mga module. Pakitignan na lang all about compound microscope para mas makita nyo yung dissected parts of the microscope. So, again, a bright field microscope. This is a microscope that allows the light rays to pass directly through the eye without being deflected by an opaque plate in the condenser. So, when you say bright field microscope, meron kang direct light source. So, from the direct light source, doon mo makikita, doon yung mga image is being projected directly using that light coming from a light source. Okay? So, it is a conventional type of instrument usually encountered by students. Ito yung student version. Pero yung sa laboratory natin is may kita mo dalawa yung kanyang ocular. Okay, dalawa yung kanyang eyepiece na tinatawag. But here, sa sample natin, ito yung mga common na nakikita sa mga biology lab, common na ginagamit ng mga estudyante. Okay? So again, kapag bright field microscope, you can see the image coming from a direct light source. So a microscope that allows the light rays to pass directly through the eye without being deflected by any intervening opaque plate in the condenser. Kaya nga tinawag siyang bright field microscope Kasi meron ka may kitang light passing through. That is the reason one that is responsible for giving you the image that you can see through the lens. So, from the eye level, meron kang second magnification lens. That is what you call the ocular or the eyepiece. And then after that, you have the first magnification level na being provided by the objective lens. Okay? So, yung objective lens natin, it can be 10 times magnification, 40 times, or 100 times magnification. So, that is the reason why you can see the specimen under the microscope. So, meron ka mga different view. Okay? Kapag bright field microscopy, una muna yung sa mata mo. And then after that, yung magnification you can find from the ocular lens or the eyepiece. Then after that, the magnification you can find from the objective lens. So, tatlo yung ginagamit mo dito para makita mo yung object under the microscope. Yung mata mo, yung lens sa eyepiece mo, and yung objective lens. Okay? Na meron siya mga different magnification. Pwedeng 10 times magnification, 40 times magnification, or 100 times magnification. So, ano ba yung mga different parts of the microscope? First is the framework. So, kapag framework, this is the structure that will support all of the parts of the microscope. Okay? So, ito yung part na meron kang arm and base, as you can see on the picture. You have there the arm, you have the base part. And then after that is the stage. Ito yung part na nilalagay mo yung microscopic slide. Nilalagay mo yung slide. Okay? Para makita mo yung object under the microscope. Siya yung ginagawang patungan ng mga slide. So, first is the mechanical stage. This is the clamping device used for holding and moving the slide around the stage. Okay? So, para magalaw mo yung slide, then, you need a mechanical stage. And then after that, yung mechanical stage, ito yung part na doon mismo pinapatong yung slide. Okay? And then ginagalaw mo siya para for you to be able to scan the object under the microscope. And then again, you have the mechanical stage control. So ito yung reason kaya gumagalaw yung mechanical stage. Okay? So it will adjust the position of the mechanical stage. And you also have the stage clip. This is the one attached to the microscope slide. Dito mo nilalagay yung microscope slide mo in order for you to keep the slide in place. Okay, so again, stage clip holds the specimen in place. And then, sa ilalim nun, meron ka dyan butas. Yung butas ang tawag dyan is the stage hole or aperture. This is the part that allows the passage of light from the light source. Okay, so from the light source, dadaan yung light doon sa butas known as the aperture. Okay? And then after that, you also have the light source or the illuminator. So ito yung sinasabi ko kanina that will provide the direct light. So the purpose of that is to illuminate the specimen. And you also have the voltage regulator. Ito yung the one that will regulate the intensity of the light. Kasi kapag sobrang bright nung ilaw, hindi mo na makikita yung object. Meron mga object kasi na kailangan nila low level of light lang. Kaya nga minsan kailangan mong i-adjust yung intensity ng light. That is the reason why you have the voltage regulator. And you also have the neutral density filter. So, minsan, pwede kang maglagay dyan ng filter sa ilalim in order for you to lower the limit of the light. Para medyo mababa yung intensity ng brightness niya. So, again, it will reduce the intensity of below the lower limit allowed by the voltage control. That is what you call the neutral density filter. So, yung nilalagay siya banda doon sa may light source. Okay? Para mabawasan natin yung intensity of the light. Another thing is the diopter adjustment ring. For diopter adjustment ring, ano ba ang kanyang purpose? They are made on the left eye by turning the curled diopter adjustment ring on the left ocular until a sharp image is seen. So, meron ka dyan sa may eyepiece mo. Okay? So, yung diopter adjustment ring na yan, ginagalaw siya to make the image more fine para mas luminaw siya on your left eye. So, nagagalaw mo yan yung ocular lens or the eyepiece para mas luminaw pa yung object under the microscope. And of course, you have the rotatable head. You have the lens system. Kapag lens system, also known as the ocular. Okay, so kapag ocular, so meron ka mga different types of lens or objectives na sinasabi natin. So yung mga lens na yun or objectives na yun, naka-attach sila sa revolving nose piece. So they have different magnification. Kapag scanner, 4 times ang magnification niya. If it is the LPO or the low power objective, you have the 10 times magnification. If it is the HPO, you have 40 times. If it is the oil immersion or the OIO, again, OIO stands for oil immersion objective. You have 100 times magnification. Hindi ko ata nasabing HPO. HPO stands for high power objective. Again, LPO, low power objective. HPO, high power objective. OIO, oil immersion objective. So, ano ba kanyang mga purpose? So, usually kapag scanner kasi, ginagamit lang siya in order for you to see kung nasa ang banda ba yung specimen doon sa slide. Okay? Kasi, of course, yung slide na yan, hindi naman yung buong slide merong specimen, di ba? Meron lang part ng slide kung saan nandun yung specimen mo mismo. Okay? Kunyari, tissue yan or blood. Nag-drop ka ng piece of blood sa slide. Okay? So, gusto mong hanapin kung saang parte ba nung slide, nandoon yung dugo. So, yun ang purpose of the scanner. Kaya nga 4 times magnification lang siya. Hindi siya ganong kalaki ang mga image na makikita mo. And then, kapag nakita mo na kung nasan yung specimen, that is not the time you're going to switch. Ilipat mo yung lens niya, yung kanyang objective. Switch mo na siya, ilipat mo na siya sa 10 times magnification. Okay? So, kapag nakita mo na yung specimen sa 10 times magnification, nakikita mo na lumilinaw na siya. Medyo lumilinaw na siya. However, yung mga details niya, yung mga minute details niya, hindi mo pa masyado makikita. That's why, you can shift, pwede mo siyang ilipat sa high power objective. So, 40 times magnification na yan, malalaki na yan. So, nakikita mo na yung mga images. Okay? So, kapag tissue ang specimen mo, kita mo na yung mga tissues. Kita mo na yung mga cells, kita mo na yung mga fibers, kita mo na yung mga structure. Okay? That is the purpose of using the HPO. Para sa naman yung OIO? Yung OIO, ginagamit ito para sa mga organism na talaga sobrang liit pa. Just like the bacteria, di ba? Mga bacteria, if you really want to see the detail of the bacteria or the detail of the cell, pwede rin naman sa mga cell, kung gusto mo makita yung kanyang detail talaga, then lilipat ka sa oil immersion objective. 100 times magnification yan. So, sobrang laki na ng mga images na makikita mo. Talagang yung details ng mga tissue, makikita mo na siya. Yung detail ng mga microorganism, makikita mo na siya. Okay? And then, yung condenser naman, it will collect and direct the light from the light source, the slide being studied. So, yung purpose ng condenser is yung nanggaling na ilaw doon sa light source, ifino-focus niya doon mismo sa slide mo. Okay? So, napaka-importante niyan kasi nga kapag yung light source mo na pumapasok sa condenser, kapag sobrang intense, sobrang linaw, hindi mo makikita yung image. Kaya nga naman, as you go along the different magnification, okay? Usually kapag scanner yan, nasa scanner ka, dapat yung condenser mo, medyo liliitan mo siya. Babawasan mo ng konti yung light. Pwede mo kasing ma-adjust yung condenser eh. So, bawasan mo na ng konti yung light, okay? Kapag nasa scanner ka, para makita mo yung image. Kapag nasa LPO ka, medyo dagdagan mo ng konting ilaw, okay? And then kapag nasa HPO ka na, dapat medyo mababa na yung ilaw dyan. Kasi kapag sobrang intense ng light, hindi mo na makikita talaga yung image na gusto mo makita. Lalo na kapag nasa oil immersion ka, konting ilaw lang ang kailangan yan. So, ginagalaw mo yung condenser. Okay? Ganun siya. Kapag nasa oil immersion na, dapat small amount of light lang ang being passed through doon sa aperture mo. Kaya nga naman, ang ginagawa mo is binabawasan mo yung light, ginagalaw mo yung condenser na yan. Okay? Next. Next is the diaphragm or the iris diaphragm. So, this is found within the condenser that regulates the amount of light that reaches the slide. So, nakikita mo yung condenser sa ilalim and then meron dyan yung structure dyan sa may condenser. That is what you call the diaphragm. So, magkasama silang dalawa. And then, of course, you have the revolving nose piece, diba? That is where the objectives are being attached. You have the focusing knobs. Yung focusing knobs, importante yan. Kasi ito yung ginagamit mo para luminaw yung item under the microscope. So, when you say coarse adjustment knob, okay, so ito yung part na yung specimen ay nilalapit mo siya doon sa mga objective. So, pag ginalaw mo yung coarse adjustment, yung lalapit talaga yung mga yung specimen doon sa objective. Okay, so bumababa yung objective papunta doon sa specimen mo or sa slide. Okay, that is what you call the coarse adjustment knob. It will initially bring the specimen into focus. And then, after the coarse adjustment knob, you have the fine adjustment knob. So, kapag lumapit na yung specimen doon sa objective, then gagalawin mo na yung fine adjustment knob in order for you to sharpen the focus para maging sharp yung mga images niya. So, again, sa normal class, ang ginagawa namin dito is, yun nga, nagdadala ako ng microscope and then yung mga estudyante ginagamit nila yung coarse adjustment and fine adjustment para maintindihan nila kung ano ba yung nangyayari kapag ginagalo yung fine and coarse adjustment. So, since wala tayong actual class, then merong video sa baba, nakalagay dyan sa module pakitignan. So, video yan regarding sa 3D ng microscope. Kung ano ba yung mga function niya and kung paano ba siya ginagalaw. Basta tandaan nyo lang, kapag coarse adjustment, the purpose of this one is to bring the specimen into focus. So, kapag ginalaw mo yung coarse adjustment, aangat yung stage. Kapag umangat yung stage, lalapit yung specimen, yung mismong slide, lalapit siya doon sa objective para maging malapit siya sa objective at makita siya. Okay? And then kapag malapit na siya sa objective, nakita mo na yung specimen, gagawin mo, magko-focus ka, kailangan mong ma-sharpen yung image. So, ginagalaw mo na yung fine adjustment knob. And then, so you have the resolution or the resolution limit or resolving power. When you say resolution or resolving power, you're actually talking about the details na makikita mo doon sa specimen na tinitingnan mo. For example, yung cell, di ba? Gaano ba kalinaw yun yung kita mo sa cell? Nakikita mo pa yung parts of the cell? Are you able to see the mitochondria? Are you able to see the chloroplast? Di ba? Are you able to see the Golgi bodies, etc.? Okay, so kapag nakikita mo na mismo yung talagang detailed parts of the cell, then doon din makikita mo yung kanyang capacity or resolving power or what you call the resolution. Okay, so when you say resolution, again, this is the extent of the detail of the magnified object. It allows two objects to be distinguished from each other. So when you say optimum resolution, this is the oil immersion. So about 0.2 micrometer means that two small objects that are 0.2 micrometer apart will be seen as separate entities. Objects closer than that will be seen as a single object. So, makikita mo talaga yung kanyang exact details. So, when you say resolution. Again, so what are the different factors that affect the resolution? First is the blue filter. For the blue filter, yung blue filter niya, nilalagay mo siya doon sa light source. Para nga naman, yung intensity ng light medyo maging mababa. Kasi kapag sobrang linaw ng light, hindi mo talaga makikita yung kanyang resolution. Mahirapan ka makita yung object under the microscope. So, yung kanyang mga details medyo malabo kasi nga sobrang lakas ng light. So, nilalagyan mo siya ng blue filter. Okay? And then, another thing is the condenser. So, sabi ko nga kanina, yung condenser na yan, tsaka diaphragm, yung dalawang yan, condenser tsaka diaphragm, you're going to adjust them in order for you to minimize pa rin yung kanyang light. Kasi kapag sobrang lakas ng light, again, hindi mo siya makikita. Hindi mo makikita yung object under the microscope. So, condenser should be kept at the highest position where it allows a maximum amount of light to enter the objective. The diaphragm should not be stooped down too much. So, hindi siya pwedeng malayo. Although stooping down improves contrast and depth, it reduces the numerical aperture naman. So, kapag sobrang layo na ng diaphragm, hindi mo na makikita yung detail under the microscope. Immersion oil should be used because the slide and between the slide and the 100 times objective. So, para saan yung paglagay ng oil? Kaya tinawag siyang oil immersion kasi nga naman, kapag ginamit mo yung objective na yan, kailangan patakal mo yung slide ng oil. Yung oil na yan, meron din siyang refractive index na tinatawag. Okay? So, kapag refractive index, ito yung factor that makes the image much more visible. Okay? So, para siyang extra lens ang kanyang purpose para maging mas visible pa yung kanyang image. Kasi kapag naglagay mo siya ng oil, dinadagdagan mo yung kanyang refractive index. Now, for the contrast naman, this is needed to make the object stand out from the background. So, kapag contrast, gumagamit ka ng mga dye or stain in order for you to see the microorganism against the background. Okay, so it is able to differentiate the different microorganism. Now, kapag low power examination, so ito yung sabi ko kanina, di ba? Una muna low power examination muna in order for you to see the specimen. So again, it enables you to explore the slide to look for the object you are planning to study. Once you have found what you are looking for, you can now proceed to the higher magnification. So, always check the condenser to see that it has been raised to the highest point to allow maximum resolution. And then manipulate the diaphragm in order for you to reduce or increase the light intensity. So, kapag ginagalaw mo yung light, nagiging sharper yung kanyang mga images. Note that as you close down the diaphragm to reduce the light intensity, the contrast improves and the depth of the field increases. Now, for the high power examination, it may be necessary to make the minor adjustment with the fine adjustment knob to sharpen up the image, but the coarse adjustment knob should not be touched. Kasi ginagalaw mo lang yung coarse adjustment knob kapag ang focus mo is the low power examination para lumapit lang yung slide. Okay? Para lumapit yung slide at makita mo yung specimen. Pero once nakita mo na yung specimen using the low power examination, then kapag nasa high power ka na, ang gagalawin mo na lang is the fine adjustment knob to make the image more sharp. So when increasing the lighting, be sure to open up diaphragm first instead of increasing the voltage on your lamp. So lagi mong gagalawin na lang sa light source mo is the diaphragm in order for you to minimize the light. Pagdating naman sa oil immersion examination, nilalagyan mo ng oil because it has the same refractive index as glass, which prevents the loss of light due to the bending of light rays as they pass through air. So yung refractive index niya, it makes the image much more sharper and clearer. Para kang merong extra dyan na lens. And then, the use of oil in this way enhances the resolving power of the microscope. So, care of the microscope, paano siya ingatan? So, for the transport, again, you have to hold it using both hands. Okay, so hold the arm in the base part. Whenever you're using the microscope, make sure that the work area is clean, walang clutter, para hindi mo masagi yung microscope. And then, of course, yung electrical cord niya, don't let the light cord on your microscope dangle. Kasi nga naman, possible na merong madapa. Kapag nahatak yung cord, possible na mahatak din yung microscope at bumagsak yung microscope. For the lens care naman, hindi basta-basta pinupunasan ang mga lens ng kung ano-anong bagay. Hindi yan pwedeng punasan ng basahan lang, di ba? Kasi yung lens na yan ng microscope, napaka-sensitive niyan. So, meron talaga tayong special paper used to clean the lens of the microscope. So, tandaan nyo, whenever you use the microscope, make sure that after using the microscope, you're able to clean the lens. Okay, so at the end of each lab session, be sure to wipe any immersion oil off the immersion lens as it has been used. Kasi yung oil na yon, pwede siyang tumigas. Kapag tumigas siya doon sa lens, definitely matigas na, di ba? Magagasgas yung lens, hindi na siya magagamit pa. Now, for the dust protection, so importante nilalagyan pa rin siya ng cover. This is used to protect the instrument during storage. Now, for the lens care, again, hindi siya basta-basta lang nabasahan na ginagamit siyaan. Meron tayong ginagamit na cleaning tissue. This is a lint-free, optically safe tissue should be used to clean the lens. So, kapag gumamit na kayo ng microscope sa lab, hingin na lang kayo ng panglinis ng lens sa lab tech natin. Hindi siya pwedeng gamitan ng tissue, hindi pwedeng gamitan ng mga basahan. Meron siya ang cleaning tissue talaga or the lint-free type. For the solvent naman, you can use soap with warm water, xylene, alcohol, acetone. So, ginagamit ito in order for you to remove the oil na tumigas or any mounting cement na ginamit mo. So, any mounting medium na ginamit mo na dumikit to sa lens or any dumi na dumikit sa lens. Tulad na namang oil na tumigas, pwede rin, di ba? Pwede mong gamitan siya ng xylene, alcohol, or acetone. For the oculars naman, you should rotate it between the thumb and forefinger as you look through the microscope. A rotating pattern is the evidence of dirt. So, once meron ka nakita dyan ng mga dirt, kailangan linisin din siya. Okay? Kasi nga naman yung ocular, kasi diba, kapag minsan nahawakan mo siya yung eyepiece, diba? So, minsan kailangan mo na siyang linisin kasi minsan may naiiwan dyan ng mga fingerprint. Objectives, again, so piece of lens tissue swab with solvents na ginagamit. For the condenser, wiping it off occasionally with lens tissue. So, lagi kayong hihingi ng mga lens tissue. Now, for the dark field microscope, this is another type of microscope being used in the laboratory. Anong kanyang difference? Ang kanyang difference is that the background is dark. Kaya tinawag na dark field microscope. As you can see on the picture, yung mga spiral na yan, that is the microorganism, and then the black is the background. So, para saan siya? This is used to observe transparent microorganism. So, since transparent nga yung microorganism, kailangan mo ng black background para makita yung microorganism. Example of this is the spirochete coming from an exudate of a syphilitic lesion. Now, for the dark field effect, it may be produced by placing a dark field stop below the regular condenser or by replacing the condenser with a special constructed one. So, para mas madali, hindi mo naman kailangan bumili pa ng bagong microscope for this one. You can actually use the bright field microscope. Kapag bright field microscope ang gamit mo, lalagyan mo lang siya ng dark film doon sa may condenser. Meron siya nilalagay na dark film. Meron akong video sa baba regarding dark field microscope. Pinapaliwanag doon kung saan mo siya nilalagay. So, pwede mo siyang ilagay doon sa may condenser, pwede rin doon sa may light source. Okay? So, meron ka nilalagay dyan na dark film. So, it is necessary to alter the light rays that approach the objective in such a way that only the oblique rays strike the objects being viewed. The obliquity of the rays may be so extreme that if no objects are in the field, the background is completely light-free. The objects in the field becomes brightly illuminated. So, kaya nagiging black yung kanyang background kasi nga tinatakpan mo yung light source niya. So, hindi siya ganun ka-intense sa kanyang light. So, kaya meron kang black background dyan. Examples are the star diaphragm, cardioid, paraboloid condensers. Ito yung ginagamit natin ng mga special condenser and special diaphragm kapag meron kang dark field microscope. Okay? Now, for the star diaphragm naman, this is being inserted into the filter slot of the condenser. Ito yung pinapatong lang doon sa condenser para gawing opaque yung it is an opaque disc in the center in order for you to block the light rays. So, pinapatong lang siya doon. That is a star diaphragm. And then, yung mga light rays mo na yan will be blocked para magkaroon ka ng black background. So, you can also use the colored celluloid stop instead of the opaque paper. This can be used. That will produce a background of blue, red, or any color. Pero kapag black talaga, usually it is the star diaphragm. For the cardioid condensers naman, the light rays entering the lower element of the condenser are reflected first off a convex mirrored surface and then off a second concave surface to produce the desired oblique rays of light. Okay, so ito yung example. You have there the condenser. You have the light stop na nilalagay mo. So, that is the star diaphragm. It allows only the peripheral light rays to pass up through the condenser. Okay? So, pag nilagyan mo ng light stop, may kita mo yung mga light rays niya is being reflected and then ang pumapasok lang is the oblique. Diba? The oblique rays. Okay? And then, again, you also have the cardioid condenser. This provides greater light concentration for oblique illumination than the star diaphragm. So, you can also use this one, the cardioid condenser. So, may kita mo dyan, it is a device mismo na nilalagay siya doon. So, hindi na siya yung normal na condenser mo. It is a new type of a condenser known as the cardioid condenser. So, yun ang kanilang difference ha. Kapag cardioid condenser, it is a new condenser. Iba siya doon sa condenser na nakikita mo doon sa compound microscope. Kapag naman star diaphragm, ginagamit mo pa rin yung condenser ng compound microscope pero may pinapasok ka lang doon na star diaphragm. Okay? Para yung light niya will be reflected. Now, for the phase contrast microscope naman, this is another type of a microscope. So, hindi mo pwedeng gamitin yung compound microscope dito. So, kapag phase contrast microscope naman, this is a microscope that will differentiate transparent protoplasmic structure without staining or killing them. So, anong difference niya doon sa dark field microscope? Kapag dark field microscope kasi, para di siya sa mga transparent na microorganism, pero those that are fixed. Kapag fixed, ibig sabihin noon, killed na yung mga microorganisms. Hindi sila gumagalaw. Okay? But, kapag phase contrast microscope, this is good for those microorganisms that are actually viable. So, this is very good for observing living protozoas and other transparent cell. So, dapat, if you want to see the movement of the viable transparent cell, mas magandang ginagamit is the phase contrast microscope. So, the phase contrast microscope is called the Zernike microscope, developed by Frederick Zernike in 1933. So, when you say contrast, the objects are categorized as amplitude and phase. When you say amplitude objects, this will show up as dark objects under the microscope because the amplitude of light rays is reduced as the rays pass through the objects. And then another thing is the phase object. These are the completely transparent one since the amplitude of light rays pass through them unchanged. So, meron kang dalawang klase na makikita under the phase contrast microscope. You can see the amplitude object. Ito yung mga dark objects mo. And also the phase objects, which are your transparent objects. So, ito yung mga makikita mo under the microscope. So, yung mga transparent and also the dark objects can be found. You call them as the amplitude object and also the phase object. Now, the different type of light rays. So, light rays that passing through the transparent object emerges either direct or diffracted rays. When you say direct rays, it means that the light rays will pass straight through unaffected by the medium. So, magmula doon sa light source, dire-diretso lang ang kanyang light rays. Okay? However, for the diffracted rays naman, the balance of the rays that are bent by either slowing through medium, rays are bent due to density differences. So, ito yung rays naman are being bent kapag yung object being observed under the microscope are too dense. Okay? So, kapag kapag dense yung object niya, then the light rays will not pass through. Nagbe-bend siya. Okay? Kaya tinawag siyang diffracted rays. Kapag naman transparent lang siya, dire-diretso lang siya, direct ray siya. Okay, kaya nga naman meron kang sa phase contrast, meron kang dark object, tsaka meron kang transparent object. Usually kasi kapag dense masyado yung object, hindi magka-pass through yung light, magbe-bend siya. Kaya siya yung nagiging dark object. Kapag naman transparent siya, dire-diretso lang yung light niya. So, kaya siya yung nagiging transparent object, diba? That you can see visible kapag gumamit ka ng phase contrast microscope. Okay, so gumagamit siya ng direct rays. Now, type of phases naman, you have the coincidence, exact phase, or the in phase. So, this is the resultant amplitude of the converged rays in the sum of two waves. So, sabi ko nga kanina, yung mga amplitude, ito yung mga dark object that you can see. So, again, kapag sinabi mong coincidence, exact phase, or in phase, so you're going to, if you have an increase in the amplitude, this will produce an increased bright object. Okay, so, yung object na yon, yung dark object na yon, so yung paligid niya nagiging bright, kaya nga naman magiging well-pronounced yung object. Okay, so makikita mo talaga siya clearly under the microscope. So, however, for the interference of the reverse space, the amplitudes cancel each other out to produce a dark object. Okay, so ito yung reason kaya nagiging dark object ng amplitude because of the coincidence, exact phase, or the in phase. So, this is the amplitude object. So, number 1, the extent to which the amplitude of light rays is diminished determines the darkness of an object in the microscopic field. Okay, so this is the image. So, sabi nga, kapag daw yung light rays niya ay nagiging nagbe-bend siya or nababawasan yung intensity of light, then nagiging dark yung object under the microscopic field. Now, for the phase objects naman, note that the retardation of light rays without amplitude diminution result in the transparent phase object. So, yung kanyang light dire-diretso lang passing through, that's why nagiging transparent siya. So, nakikita mo siya clearly. Okay, so these are the different type of rays. So, you have the direct and the diffracted rays. So, kapag direct ray, dire-diretso lang siya doon mismo sa image. Kapag diffracted, may kita mo meron siya mga bending of the light. So, a light ray passing through a slit or transparent object emerges as a direct ray with several orders of diffracted rays. The diffracted rays are 1/4 wavelength out of phase with the direct ray. Now, for the coincidence and interference phase naman, again, meron kang tawag na in phase and you also have the reverse phase. So, note that when two light rays are in phase, they will unite to produce amplitude summation. Light rays in reverse phase, however, cancel each other to produce dark objects. So, ito yung reason that's why you can distinguish between the light and the dark object. Kasi kapag reverse phase, it will produce the dark object. Kapag naman in phase, it will produce the clear object or the phase object. Okay, so that's why you can distinguish both the dark and the light images because of the coincidence and the interference. Now, this is the parts of Azer night microscope. Okay, so you have the annular, marami siyang parts, but the important part are the following. You have the annular stop, this one. This is found within the diaphragm that allows a hollow cone of light rays to pass up through the condenser to the object on the slide. So, ito yung reason kung bakit yung light ray na direct ay pumapasok doon because of the annular stop. Now, for the faceplate naman, this is a special optical disc located at the rear focal plane of the objective. So again, kapag faceplate, importante siya to see the transparent object. Now for the face ring, this is found above the faceplate that advances or retards the direct light rays. So yung direct light, it will pass through the annular stop. After that, it will pass through the faceplate. Then you have there the face ring. Again, the purpose of this one is to advance or retard the direct light rays. So, ginagamit mo siya to adjust the intensity of light. Now, for the fluorescence microscope, so this is very similar to the dark field microscope, yung fluorescence microscope mo. However, ang ginagamit mo is that you're going to add the fluorescent dye here, what you call the fluorochromes. Kapag fluorochromes na yan, these are dye being added to the specimen that will produce a fluorescence. So this is used to observe microorganism in a fluorescent stage. Okay, so nilalagyan mo siya ng mga fluorescent dye para yung magiging kulay niya tends to fluoresce. Example of different fluorescent dye are auramine O, acridine orange, auramine rhodamine, chalcofluorite, fluorescent isothiocyanate, or the FITC. When you say fluorescence, so this is whenever a physical body absorbs energy, as in the case of the activated molecule, the energy doesn't just disappear, it must reappear again in some other form. So yun ang purpose of the fluorescent dye natin. So yung fluorescence na yun, it is an energy being emitted. And then that emitted energy, you can observe under the microscope. So this new manifestation of energy may be in the form of chemical reaction, heat, or light. Fluorescence is initiated when a molecule absorbs energy from passing a wave of light. The excited molecule, after a brief period of time, will return to its fundamental energy state after emitting fluorescent light. Again, kapag fluorescence na yan, you're going to observe the energy being emitted as it passes a wave of light. Okay, so again, it is affected by pH, molecular structure, and temperature. So this is the parts of a fluorescent microscope. First, you have the mercury vapor arc lamp. This serves as your light source. And then, so after that, you also have the heat filter. So yung heat filter na yan, this is used to remove infrared rays generated by the mercury vapor arc lamp. So yung kanyang light source or the mercury vapor arc lamp, this is also the one responsible to supply the light. Besides the light, ito rin yung nagpo-provide ng heat, 'di ba? Kasi kumukuha siya ng heat coming from the light. So this is preferred over incandescent one because it produces an ample supply of shorter wavelength of light. So UV, violet, and blue light. Yun ang makikita mo sa mercury vapor arc lamp or the light source. Then after that, it will pass through the heat filter to remove the infrared rays or infrared rays light generated by the mercury vapor arc lamp. Then after that, it will pass through the exciter filter. This will absorb all of the wavelength except the short ones needed to excite the fluorochrome on the slide. Okay? And then after that, so they are very dark and are designed to let only the green, blue, violet, or UV rays. So ang nakakapasok lang dyan is the green light, blue light, violet light, or the UV rays. Okay? Yun lang ang pwedeng pumasok. After that, it will go to the dark field condenser. So para din siyang, ang purpose niya is just like your condenser sa compound microscope, di ba? So ang purpose niya is to regulate all of those light passing through. So this is used to achieve best contrast of the fluorescent object. Majority of the UV light rays are deflected by the condenser, protecting the observer's eye. Kasi nga naman kapag papasukin mo yung UV rays doon, malamang masakit siya sa mata. So, pwede mag-cause ng damage sa eyes. So, kailangan, ang purpose of the dark field condenser is to control the intensity of light. Besides that, this will now tend to remove or deflect all of the UV light. And then after that is the barrier filter. Now, the filter is situated between the objective and the eyepiece to remove all remnants of the exciting light so that only the fluorescence is being seen. Okay, so ito na yung last part kung saan dumadaan yung light na kailangan no sample. So, only the light that will tend to excite the molecules is being passed through sa barrier filter. Okay, so kapag na-excite na yung kanyang mga molecules, then lalabas na yung kanyang fluorescence. Now, after that is the electron microscope. So, this is a very special microscope. This can magnify the objects up to 100 times magnification. That's why, pati yung mga viruses, makikita mo na dito, yung mga sobrang malilit na microorganism, yung mga details mismo, kitang-kita mo under the electron microscope. So, you have two types. You have the transmission and also the scanning. Kapag transmission, this will view the internal structure of the microorganism. So, yung pinakaloob-loob ng microorganism, makikita mo na. Kapag scanning na yan, surface lang yan. So, yung surface ng microorganism makikita mo, you can see it in a 3D. Okay? So, yung 3D na scale, makikita mo siya kapag scanning electron microscope. So, surface lang talaga. Pero kapag gusto mo makita yung kanyang details, then you can see the internal structure. That is what you call the transmission electron microscope. So, meron akong video sa baba ng module pakitingnan regarding electron microscope. So, para makita nyo kung paano ba talaga yung kanyang mga magnification. So, again, electron microscope, this is used for research purposes. So, hindi talaga siya common na nakita sa laboratory. Sa Philippines, meron tayong mga, I think, tatlong electron microscope sa Philippines. Yung isa nasa RITM, yung isa nasa St. Luke's, yung isa naman nasa UPPGH. So, tatlo lang ang microscope natin sa Philippines na electron. Okay? Kasi nga, for research purposes lang siya. This is for special purpose. So, that is the lesson. Please make sure to view the other videos regarding the microscope para mas makita nyo kung paano ba talaga nakikita yung mga objects under the microscope. Kasi nga, wala tayong lab, 'di ba? So, dapat, usually kasi kapag may klase tayo regarding microscope, pinapagamit ko yung microscope sa mga estudyante para mas maintindihan nila kung paano gumagana yung microscope. Pero since wala tayo, you can watch the video as a supplement.